Hello, everyone. Welcome to today's live broadcast, Cross-Validation of Antibodies using DigiWest, presented by Drs. Markus Templin and Anthony Couvillon. I'm Christina Jewell of LabRoots, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar presented by LabRoots and sponsored by Cell Signaling Technology. Cell Signaling Technology, CST, is a private family-owned company founded by scientists and dedicated to providing the world's highest quality, innovative research and diagnostic products to accelerate biological understanding and enable personalized medicine. For more information, go to www.cellsignal.com. Here's how the presentation works. We want to hear from you. Questions, comments, and even answers can be submitted via the green Q&A button at the lower left of your screen. We'll try to get to everyone, but if not, we'll make sure to follow up with you by email. You can enlarge the slide window by clicking on the screen icon in the lower right-hand corner of the slide window. If you can't hear or see this presentation properly, let us know by clicking on the support button at the top right or the Q&A button in the lower left. We have two speakers today, Dr. Markus Templin, Head of Assay Development, and Dr. Anthony Couvillon, Scientific Marketing Project Manager at Cell Signaling Technology. This two-part webinar will focus on the use of novel methodologies being developed to challenge the specificity of research antibodies and how those technologies apply to antibody producers such as Cell Signaling Technology. Our first speaker today is Dr. Markus Templin. Dr. Templin heads the Department of Assay Development at the NMI, Natural and Medical Sciences Institute at the University of Tübingen. In a multidisciplinary environment, scientists from applied physics, chemistry, biology, and biochemistry are working together in his department to develop advanced assay systems for industrial clients. Markus Templin's research is focused on the application of microarrays for proteomic and cancer research, and he holds several patents in this area. He studied biology at the University of Tübingen, did his postdoctoral research at the Edinburgh University of Scotland and the Max Planck Institute for Developmental Biology, Germany, before joining the Natural and Medical Sciences Institute. I will now turn the presentation over to Dr. Templin. Dr. Templin?
Hello. It is a pleasure to talk about some recent work we did in our Tübingen lab, and it is about the development of a novel method, a variation of the Western blot. I will go into some detail of that, but more important, I think, for this seminar, a first application, no, not the first application, but an early application, which really is classification and characterization of antibodies. Very briefly, to my background, so it was mentioned that I'm working at the NMI at the University of Tübingen. Interestingly enough, it is not part of university, the NMI, but an independent organization, a foundation, and we're bringing research into use, so focus is applied research. The institute is shifting quite a lot into biology, and we're actually working as a CRO at the moment, doing work with assay systems we have developed. But that's enough for an introduction. I would like to go what did interest me over the last 15 years and how we came to use technologies we have in-house to develop a new variation of the Western blot. So you see here a slide from the CST website which shows the MAPK signaling pathway. It is a complicated thing, a lot of proteins, and all of these proteins come in different flavors. They can be activated, which very often means phosphorylated, and detecting phosphorylation tells you a lot about the state of activity of a pathway inside the cell, which determines the behavior of the cell. To get an idea of what really is happening inside the cell, you want to measure these modification and maybe also the absolute amount of proteins. And for that, there are different methods. Mass spec has become very important, but the classical way, and still probably the most sensitive way, is looking at proteins using the classical Western blot. And for that, you need antibodies. So you get bands, and if you get a good antibody, you get an idea of what really happens. What you see here is just a few bands from a small and simple experiment, which, by the way, was still quite a bit of work, so several days of work. But if you now think about going into much more detail, you need much more antibodies, and then work really starts to get big, and the amount of material you require is growing and growing. So we were thinking about how to get the classical Western blot into a format where it can accommodate more and more incubations. And we started, and we wanted to do that with a very classical way of separating denatured proteins in an SDS-PAGE gel, and this is what you see here. The Western blot then requires the transfer of the separated proteins onto a membrane, which should be shown here in the next part of the picture. So you've got a blotting membrane, which is usually incubated with an antibody to give a picture of a band which has been detected by this antibody. Now things start to get a bit different. We're not looking at whole gels, we're starting to look at only one lane, which is one sample. And this one sample shows all the proteins which are present in the sample we use to generate fractions. And interestingly, these fractions are somehow size fractions. So we cut the band from top to the bottom into small stripes of membrane. In these stripes of membrane, actually we cut 96 different stripes out of one lane, have now the equivalent of 96 size fractions. With this material, we go into a microtiter plate. There comes the 96 from. And each of the snippets we have is put into one well of the microtiter plate. So each of the wells really contains one defined size fraction of proteins. We can now elute all the proteins from the stripes. That's part of a patent we have on this method. And we add beads to each of these wells in the 96 well plates. Interestingly, there is technology available, and we use the Luminex technology for that, that each well gets a different colored bead. So in a way, you can now transfer the proteins which were in solutions, came from the strips, onto the surface of the beads. And since the beads are 96 different flavors, 96 different colors, you have now the transfer of the side fractions onto distinguishable beads, which means you get, when you pull off all of these, a section of beads which are still identifiable. So yellow beads might carry proteins of the size of 60 kilodaltons. Blue beads might carry proteins exclusively of the size of 18 to 20 kilodaltons. So quite a bit of work what we have done to that point, and what we end up with is kind of a Western blot membrane in an Eppendorf cup. What's the advantage of this amount of work? It's very simple. The amounts of beads we do have here is sufficient to run assays, not one assay as in the classical Western blot, but quite a few assays, and we run 200, 400, or even 600 antibody incubations with just one of these Eppendorf cups for one sample. So instead of having just one assay, maybe two antibodies, maybe three antibodies on one sample lane, you can now have 100-fold the amount of assays running from this amount of material, which is 10 to 20 microgram, what we classically use. So how does it work? So we take aliquots, let's say a fraction of a percent, into a 96 well plate again, and to each of the wells we put in a different antibody. What happens now is the antibody does recognize beads which carry the right protein. So for each of the wells you get 96 different signals, and these 96 different signals can be plotted according to size. And what you see, and this is indicated here, is you get kind of a chromatogram, and this chromatogram gives you information on, first of all, where signal is detected and what size the beads carry proteins of. So let's make that a bit clearer, and for that I prepared a slide which is shown in the next one. So we do have, if you do run a classical Western blot, a stripe where all the proteins which have been separated are shown. Here we did two stripes of these Western blots, and we're using two different antibodies, recognizing a protein which runs at 38 kilodalton, the AF2 alpha protein. We've got two different antibodies, one recognizing whole protein and one recognizing a phosphorylated version of the protein. What you actually see is, yes, there is a band. This band is specific. It comes at the right place. We think so. And you see the same band for the phosphorylated version at the same place. And again, the molecular weight is the same, indicating that they're really both the same protein, one time the phosphorylated version, the other time the total version of the protein. If you now take this separated lane, you can cut it into 96 different size fractions, as I explained before. And what you see in the next slide is small dots where the membrane was cut. And when you now incubate the beads with the antibody, you see signal occurring at the snippets, which were carrying the 38 kilodalton fractions. And indeed, there is a peak occurring for both of the versions of the protein. You also see some sidebands, and you see that in both the classical Western blot and in the DigiWest diagram representation we have there. You now can use the numeric data which was generated by the Luminex machine and make a picture back out of this data again, and this is seen in the next slide, and that looks very much like the initial Western blot. So in a way, that's a very simple way of getting the information you usually get from a Western blot as numerical data. And as I said, the big advantage is instead of running one, you can run hundreds of antibodies on this material you use for your separation in the SDS-PAGE. So to make that a bit more clear, the next slide shows you one antibody, and that's just a microtiter plate full of incubations with the Western DigiWest beads we were making. So, okay, it looks nice, but what is it? What does it mean? Is the sensitivity good enough? Is it linear? And how good is reproducibility? And for that, I will show an example, and this example is a very classical example. We used the Lycor system, which is still one of the best readouts for Western blotting, to incubate a Western blot in which we used a cell line. It was a HapG2 cell line in that case, where we spiked differing amounts of the fusion protein of the ERK2 protein too. So the differing amounts were really spanning 3 orders of magnitude, from 20 nanograms spiked in to 20 picogram. And what you see is 3 bands in the classical Western blot. It's the 2 intrinsic ERK bands, ERK1 and ERK2, and also the fusion protein, which is bigger, which we spiked in. You now can use the picture from the Lycor and quantify, and you get numeric data out of that. So we did do the same thing again, and we're processing the samples in the DigiWest. And what we got is the DigiWest diagrams. And you see just one here on the top. Again, it is the same thing. You've got a big band, which is the lower band from the 2 intrinsic one. You get a lower intensity band, which is the second one. So you see the ERK1 and 2. And at 74 kilodalton, you do see the fusion protein with a certain amount, and that is for the 1.25 nanograms. You can do that for all the other lanes, and you see that the intensity drops if you spike in lower material, but it really goes down to the 20 picogram. There's still a clear-cut peak seen. The same is true if you go up to higher amounts, the 20 nanograms, so the signal gets very high, so we put an inset to really show that. So you really see that over 3 orders of magnitude, you get peaks, and you can actually quantify these peaks. And if you do that, if you quantify the peaks from the different lanes here used by the DigiWest, you get the diagram, and what you really see on the right-hand side is there's a very good linearity. What you don't see is these measurements were done in triplicate, so the variation is low, and we usually get an inter-assay CV which is well below 10%. So if you do the same thing with the Lycor system, it is not better, not worse. It is actually fairly similar. So what we do have here is a technology, the DigiWest, which is, when looking at linearity and sensitivity, on par with the Lycor technology. But keep in mind, the same amount of material you would need with the classical Western blot is now sufficient for hundreds of assays. Okay, that is good. We really like that. We developed the method further, and it is now really in the state that we do offer that as a service. But if you do have the possibility of running hundreds of assays in parallel, you need to have the hundreds of antibodies. And for that, we started, and that is actually started approximately 10 years ago, ways of qualifying the antibodies we're usually buying. And we buy a lot of antibodies, so our fridge is filled with more than 1,500 antibodies. And what we started with is a very classical way of qualifying our antibodies for our use. So we were still working a lot with mouse material, mouse tumors, mouse tumor cell lines, but more often we're actually working with human material, and that is human cell lines, human tumor cell lines. Human primary tissue has become very, very important. So we set up a set of materials we used for qualifying our antibodies. So what you see here is 3 different mouse samples, which is normal liver from mouse, and 2 different types of tumors, mouse tumor, liver tumors, where we were running classical Western blots. And what you see here are small stripes which we cut out of our Western blot. The same is true for the human material. Again, a set of human cell lines. What you see here is 5 and 2 primary tissues, 1 tumor tissue and 1 normal tissue. If you do that, you add an antibody, and that's really a very, very good antibody, 1 monoclonal rabbit we purchased from CST, which gives you what you really want to have from an antibody. And we see here that the antibody is working on all the materials, the mouse materials and the human materials. And this antibody we qualify as a good working antibody, and that made it to our collection. We did this kind of analysis for 600 something antibodies, so we do have quite a big basis of Western blot qualified antibodies. But now when we had our DigiWest available, we were thinking, okay, is there maybe a quicker, faster, simpler, and maybe even better way of getting an idea if the antibody is working on the different materials we are interested in and we have on hand. And we use now the DigiWest for qualifying our antibodies, and that is seen in the next slide. Looks very similar, and what you see is again 4 of the samples which we are using for the human samples, which we're using for qualifying our human antibodies by classical Western blot. Again, the same thing. Some of the mouse samples we've got quite a bit of information on. It is 4 now. But we added here different material we're interested in. It is rat kidney, it is rat liver. We also have dog kidney and liver, monkey kidney and liver, and mini pig kidney and liver. So this really points to one of our areas of our expertise. We're doing toxicology and toxicology assays using the DigiWest. So what you see, again, an antibody from CST, a good antibody, a monoclonal rabbit, which gives a perfect band at human and mouse. And if you now go to rat, the band is very weak, not unspecific, but it is weak. Dog, monkey, and mini pig is also giving the right band at the right place. So what you do have here now is well beyond what we had with the classical Western blot. Information on the performance of this antibody in different species, and there also in different types of tissue in this species. I'll put a second example on that. Again, an antibody against the kinase, which is at that time not from CST, which gives some sidebands, which interestingly doesn't give any reactivity in rat. Very poor and not really any reactivity in dog, but it is working again in monkey and mini pig, but there it's have a sidebands. So we've got now the idea how the antibodies work on different species. To show it in a more systematic thing, in a more systematic view, we did that with the 1,200 something antibodies we're using regularly right at the moment. It is, when you go to human, a good fraction of antibodies which doesn't give any sidebands on any of the observed materials. So there is also a larger fraction of the antibodies, which is a quarter of all we tested roughly, which do give some sidebands in some tissues, but they are perfectly usable. And since you do have the information from the size separation using the DigiWest, they're really no problem to use. There's quite a big section of proteins where we don't get any signal, which is perfectly possible. So not everything is expressed in any tissue. And if you're thinking about phosphorylation, this really might be very important because some proteins are only phosphorylated under certain circumstances. Critical it gets with other things where the size is not there or the signal is really poor. So to summarize, it is a fast approach to look at quite a few antibodies. The sample consumption is very low for all of this. What I was showing here, you're thinking really on maybe 200 microgram of material which is needed, and you get this information relatively quick, and you can use dozens of samples, be it different tissues, be it different species, be it different cell lines, and they can all screened in parallel. Okay, so much for the antibody validation, but I was talking about the DigiWest, and the DigiWest, so same thing, well, it was a bit too quick, same thing on the millipig, and what you see here, that from roughly 700 we tested, some 60% are working, which is interesting because now we have a collection, a validated collection of antibodies, which can be used in material which has been created or get from mini pigs. Okay, but to summarize that, you do have the possibility, and we do have a set of 700 to 800 antibodies for mouse and human material, which is about a number which really makes sense if you can run hundreds of assays on the DigiWest. If you go to the species which might be more interesting for pharmacology, toxicology, rat, cynomolgus, dog, and mini pig, we're still well beyond 300 for all of these species. And you can compare the quality of the antibody by that relatively easily. Okay, but I would now like to go back to the DigiWest, and just to summarize, we can use all types of materials, be it cells from cell culture, be it tissues, be it tumors from tumor models, and run them on a classical SDS-PAGE, blot them as classical, transfer all of the proteins which were in the sample onto beads, pool the beads, and use the bead pool as equivalent of Western blot membranes and generate numbers, bands, which we can then use for getting larger data sets, which are not really proteomic data sets, but they're not too far away from that in bigger protein profiling experiments. The method is finally getting published. It is in press, and if things go right, it will be very soon, maybe even tomorrow, that it will go online. Okay, so in the paper we will publish, we have also some examples how the DigiWest can be used. And one thing we're interested in my lab is resistance development. In the collaboration with Bernhard Küster from the TU in Munich, one of the big guys in mass spec, we designed an experiment where material, a cell line which was designed in Bernhard's lab, which was rendered resistant against lapatinib, was compared to the parental cell line. So the cells were grown over a long period of time, month, half a year nearly, in the presence of increasing amount of lapatinib, and they learned to deal with that. And we took lysate from the cells and analyzed that by a DigiWest, and we took lysate from the parental strain and analyzed by DigiWest. So it is simply on comparative analysis. At the same time, or actually before we did that, Bernhard or Ben were analyzing these cells again. They used an enrichment of kinases, a method which they developed in their lab, the kinobeads, so that only the kinases were pulled out of the cell lysates. And what they then did, they compared the amount of the kinases they fished, and it's essentially all kinases present in the cell lines, and they made ratios out of the resistance over the parental strain. That's shown in the next slide. So there is a set of 24 kinases which are different between the strains. A set like the Ephrin-A4 is upregulated under resistant conditions. Another set like Ephrin-A2 is downregulated. And what you see here is simply the ratio as a log 2 is there, and what you see, you get upregulation up to a factor of 4 and downregulation up to a factor of 10. Interestingly, we did the same experiment, but not using mass spec, but by using our DigiWest. And what you see, here in blue is the ratio we calculated from the DigiWest experiment. And interestingly, it is very close to each other. So in a way, we not only have a good correlation between the DigiWest and the mass spec data, but we get somehow a benchmark, a validation that the DigiWest experiment could give you similar information as the mass spec approach. A nice thing, and that came to our minds when we were doing these experiments, is we don't need to purify the kinases out of the crude lysates. We can use the crude lysates directly, and that gives the possibility not only to look at the kinases, and what you see here in the next picture is what we got out of the kinase enrichment, but also to look at the whole lysates. And with the whole lysates, you as well get the possibility to look at kinases. The RSK1 is well visible over there, but you also see different things. And what was interesting for us and for our work was that you see the counteracting proteins, the dual specific kinases of a lot of the dual specific phosphatases, a lot of, well, connected with MAP kinase signaling, but getting rid of the phosphate occurring here as well. So the information goes well beyond the kinome. It gives you the possibility to also look at other proteins, and here the phosphatases were especially interesting. Another type of protein which was especially interesting was the P52, and our analysis of the data set we had, and we were using more than 200 antibodies on that, was pointing to a role in resistance development for P53. And we did an ingenuity plot, an ingenuity analysis, and when you look at the amount of P53, you really see that the resistant strain had a strongly downregulated amount of P53, which is an indication what really happens in this cell. So this was a first example which somehow validated the approach, but also really gave new information. Very briefly, on two other things I would like to go, was mentioning the importance of resistance in our lab, and we are also looking at primary material, primary tumor material, which we got from the clinic here in Tübingen on changes in the kinome, not only, but also in other proteins that are observed within primary ovarian cancer. So resistance, I won't go into detail in that, is a big problem when treating ovarian cancer, and it's really not quite clear when and how the resistance will occur. And what we had was in retrospective studies, so we had material from patients which showed relapse after the operation, and we had the initial tumor material which was removed from the patients, and we were looking at these type of material from patients which had a relapse and patients which were cured after half a year to 3 years time. We did the same thing. We tested, first of all, a lot of antibodies. It was more than 1,200. We chose 466 antibodies, and we're looking for, in a smaller set, that was 24 in that case, for differences in expression or in activation. That is briefly seen here. You get so much material, so much data that the classical heat maps from mRNA expression are the only way to visualize them. And if you condense that down, we ended up with a smaller set, so around 10 proteins, which really discriminated material, cancer tissues which developed resistance later on from the material which stayed sensitive to the platinum, which was used as a treatment. And interestingly enough, it was exclusively, mainly phosphorylated versions of protein which we found on that side. That work is still ongoing and will be published sometime soon. And the indication we do have that MAPK signaling is important, NF-kappa B signaling is involved, and also mitosis and proliferation are changed between the sensitives and the insensitive tumors. Okay. Last example I would really want to give is that you really can use the system with not a lot of material. So I showed you can use that with cell lines. I showed you can use that with primary material, well, that was primary tumors. But what we did as a last way of really looking how good the method is, we were taking sections which had been prepared from tumors. In that case, it was mammocarcinoma, and we had a look at the pathologist had a look at the tumors which were found in the sections, which were either invasive ductal carcinoma or very often directly adjacent to the invasive ductal carcinoma, the ductal carcinoma in situ. So the supposed preform you find fairly frequently in the sections. By using laser capture microdissections, we were capable of isolating cells of one type and also cells from the other type, from one patient. So it really is a matched way of comparing the type of tumors which were found beside each other. The number of cells found was relatively low, just a few thousand, which we could collect by laser capture, and the amount of material you get out of that is in the range of a microgram, maybe a bit more, maybe a bit less. And there we tried if it is possible to run a DigiWest with this low amount of material. And actually, yes, it is possible. So 160 incubations we did, and we get really differences. So you see here the invasive versus the in situ carcinoma, some proteins, and as examples, some obvious things like the cytokeratins. The cytokeratin 18 is unchanged, 19 is getting down in the invasive form. On the other hand, 14 is going up in the invasive form. So to summarize very briefly, so we did a match pair analysis from laser capture microdissected cells we got from the slides directly. We had not a lot of material, so range of a microgram, and we were able to test 160 antibodies on that, and we have quite a comprehensive information on pathway activation and phosphorylation. So that brings me to the end. So I was talking quite a bit about a novel technology, as I mentioned, to be published very soon. It is a version of the Western blot which starts with classical protein separation in an SDS-PAGE and blotting to a membrane, then transfer to beads, which are in our case read out with the Luminex system. And for that you can really use what you usually put onto a gel, 15, 20 micrograms, to run hundreds of different assays. For that, we needed qualified antibodies, and part of my talk was really how we qualify these antibodies these days. And for that, we built, well, we call it a streamlined antibody characterizing platform. We still rely on the information which come from the companies, but if you really look at different types of material, different tissues, it is very useful to have an idea if the antibody we want to use is working or is not using. So at the end, 3 examples how you can use the DigiWest. So there is a broad range of applications in translational research. And very briefly, very important, these are the key people which were involved in the work. So Fridolin started that as a PhD student, and all the others in the group are still heavily working with that. And maybe quite interesting, after a while we found out that the method is not only working and is not only giving good results, but really fits into our portfolio of commercialized assays. So I was mentioning that the NMI and the NMITT is working as a CRO, and we now do offer DigiWest services, mainly performed in our Berlin site. And if you're interested in that, there you see the webpage where you can find more information. Okay, and that's it from me. Thanks for listening.
Thank you, Dr. Templin. Our next speaker is Dr. Anthony Couvillon. Dr. Couvillon attended Colby College in Waterville, Maine, USA, where he majored in biology and chemistry. Post-graduation, he became a research associate in the laboratories of Dr. Christopher L. Carpenter and Louis Cantley at the Harvard Medical School, where he was introduced to the fascinating world of signal transduction, lipid kinases, and the role of RAS-like proteins in disease. Under the tutelage of Dr. John H. Exton at Vanderbilt University in Nashville, Tennessee, Dr. Couvillon received his PhD in molecular physiology and biophysics while studying the role of Rho family proteins and their regulators in neuronal function. He then returned to Harvard and Beth Israel Deaconess Medical Center, where he rejoined the Cantley lab to continue his work on Rho family GTP-ACEs and their role in immune cell development and function, neuronal plasticity, and cardiac development. Dr. Couvillon joined cell signaling technology in 2009 as a development scientist, developing and validating antibody products in the MAPK, lipid signaling, and other pathways. He has also worked extensively in the proteomics space, developing novel antibody reagents against novel post-translational modification and the reagents that detect specific substrate motifs for high-throughput proteomics analysis. Dr. Couvillon currently works as the scientific project manager focused on initiatives promoting antibody reproducibility in research. I will now turn the presentation over to Dr. Anthony Couvillon. Dr. Couvillon?
Thank you, and welcome everybody to the webinar. There are two main points that I'd like to talk about today in following up to Markus's presentation, and that is, why do some antibodies, like those from CST, work well on a platform like DigiWest? And the second point is, what is the importance of cross-validation on a platform such as this? And if there's one message that I hope that you take away from this part of the webinar, it's that all antibodies must be validated in the application that you're using. Just because an antibody is validated in one application does not mean it will work well in another. Most reputable antibody suppliers will often validate their antibodies in a set of core applications, such as Western blot, immunoprecipitation, immunohistochemistry, immunocytochemistry, flow cytometry, and/or CHIP. Of course, the applications tested depend on the targets in the antibody. For example, you wouldn't necessarily consider testing an EGF receptor antibody for CHIP. However, testing an antibody application does not mean that it is validated. For every application, the specificity and sensitivity of the antibody must be determined. The process of properly validating an antibody is long and complex and relies on the use of the right controls, cells and tissues that differentially express the target of interest, knockout models, CRISPR and siRNA, choosing the correct model systems, and validation and use of agonists and inhibitors. The most important factor in our hands is cross-validation, that is, testing and validating the antibody in multiple applications, such as those that Markus has just described. This information provides confidence in the specificity and sensitivity of the antibody. Most vendors will indicate which applications have been tested, but not necessarily validated for their products. Customers and end users should always review the data to see if the vendor has validated an antibody in the application you're using. If not, you'll need to do it yourself. Again, reputable vendors will often indicate the applications that have been validated for the antibody and provide both the data and the protocols to support this result. In addition to the core applications I just described, CST often cross-validates its antibodies in a number of additional applications to prove specificity and functionality. These include the use of ChIP-seq, MEDIP, peptide arrays, competitive ELISAs, DNA dot blots, in-cell westerns, multiplex IHC, etc. Multi-tissue arrays, I've illustrated a few of those here. The applications are used to cross-validate are highly dependent on the antibody target and the desired functionality. Oftentimes, validation by multiple applications, either internally or externally, can increase your confidence in the specificity of the antibody, but please remember, functionality in one assay does not predict efficacy in another. One of the reasons that we at CST get very excited about platforms like DigiWest is seeing our products tested and validated in another application. Many of our products are used widely in other novel assays, such as nanostring and mass spec imaging techniques, such as those being developed by IonPath. There's a paper here that you can reference for that information. We get excited because generally our antibodies perform well in these systems due to the high level of validation that we do in-house, and it's nice to see that confirmed by external users. Are all of our antibodies perfect? No. but it does not mean that the antibody isn't specific. It may mean that the antibody does not work in this particular assay. And typically, when we find a publication such as this, we still review all the data and revalidate the antibody just to be sure. So why is independent validation in every application so critical? And I'm sharing with you here an example of a product that I developed a few years ago. These are Western blots from an animal immunized with HSP70 antigens. And this was an antigen that we knew to perform well by immunofluorescence and immunohistochemistry previously. And so we needed to develop a new antibody that had multiple applications. So we isolated antibody clones from one rabbit and tested them here by Western blot. I'm showing you 4 representative clones. Keep in mind that all 4 of the clones I'm showing you here are from the same exact rabbit and the same peptide. We tested specificity independently. I'm just not showing it here. One of the desired applications was immunofluorescence. So we tested the same 4 clones, again, that looked identical by Western blot, and came up with a situation in which we had very different localizations. We went on to further validate using heat shock and other methods to alter the location of HSP70 in the cells, but none of the clones proved to be correct. So this is one of many examples that we've seen of how an antibody looks good in one application but performs poorly in another. Again, this illustrates the need for application-specific validation, including all of the techniques that I've mentioned previously, and I've reiterated those here. But one that's often overlooked is the use of a correct protocol. So why are correct protocols so critical in antibody development? How often are the methods sections in papers detailed enough for you to be able to replicate an experiment done in another lab? I'd argue that this is not very frequent. The same is true for antibody suppliers. At CSG, we try multiple protocols when testing our antibodies, and when we recommend an antibody for a particular application, we not only provide the data, but also a detailed protocol with the steps and reagents used so you can repeat the experiment in your lab. This is important because different fixation and permeabilization conditions can adversely affect your results. As you can see here, just changing the permeabilization condition from triton to glucoside made a huge difference in the correct staining of our BCL-XL antibody, and that's the protocol that appears on our website for that product. Sometimes the results are not so subtle, and the buffer conditions can make a difference between complete success and complete failure. At CST, we are committed to your success, and we provide the specific protocols used in order to validate the antibody. As always, if there's a question about whether an antibody works in a particular application or whether it's been tested or the conditions under which it works, we encourage you to call our tech support so they can help you. Any of these conditions can adversely affect the outcome of an experiment. Often overlooked is the quality of the sample, how long it's been stored, the buffer it's in, the fixation and permeabilization time, the freshness of buffers obviously has a huge impact. And again, I will argue that the vendor should absolutely provide a detailed protocol for you to use when you're using one of their products to remove these variables and make your experiments easier to carry out and interpret. In closing, CST's validation principles illustrated here are specificity, sensitivity, quality, authenticity, methodology, and transparency. And all of these things lead to greater reproducibility for you and for all academic and industrial research. If you're interested in learning more, please visit our website or log in and sign up for the next LabRoots webinar, not next, but the webinar that I'll be presenting in October, which will get into this and the role that antibodies play in the entire reproducibility crisis coming up in October. And with that, I will say that, again, if you need assistance with an antibody or you have questions about this, our validation procedures, please call us, let us know. We'd be happy to hear from you. And with that, I will close, and Markus and I will take questions.
Thank you, Dr. Couvillon, for that informative presentation. Okay, we want to get right to your questions and input, so here's a reminder how to let us know what you have to say. Questions, etc., can be submitted via the Q&A button at the lower left. We will try to get to all of you, but if not, we will be sure to follow up with you after the broadcast. Okay, our first question is a two-part question. Is the membrane manually cut into 96 slices or automated? And how do you accurately assign molecular weight to these 96 slices?
Okay, well, that's actually a nice question. So first of all, the automation is there. So there are devices which are called cutting plotters, which we use for cutting the membranes. They're very accurate. So they usually use the same technology as inkjet plotters to position the knives. So it's very simple to get equally distant pieces of the membrane. And in the paper, we will go into that also in some detail. The second question is actually a very interesting question. While we started with running an extra lane with a marker, and we're looking where the markers were running and then simply transferring the distance to the snippets we generated, we can do it in a totally different way. So the DigiWest is capable of running hundreds of assays. So hundreds of antibodies are usually used during one assay. So now, for each of the antibodies, you know that the protein which is to be recognized has a certain molecular weight. So what we usually do is pick something like 15 of these proteins which are detected by the DigiWest, knowing their molecular weight, knowing on which sniplet, on which bead they were running, and using them as an internal marker. This really has got the advantage that each and every separation, which is obviously dependent also on the SDS-PAGE, will give you the actual size of the protein, which you know where it is, and the accuracy of this kind of assigning the molecular weight is very, very good. And again, we describe that also in the paper in some detail.
Thank you, Dr. Templin. Next question. How do you ensure reliability of results over time? Meaning, when do you need to use new batches of antibodies, mainly for polyclonal?
Yeah, so I can take that question, and that's a very, very good question. The way that we, and I can only speak for CST, the way that we ensure reliability, consistency over time is by validating every lot of a new product or of a product in every application for which it's approved. And it's very important that you formulate the antibody in a way so that it works exactly the same from lot to lot to lot. Now, this is particularly difficult for polyclonal antibodies because there is generally drift in polyclonal antibodies over time. And so at CST, we're very, very, very careful to ensure that our products are consistent from lot to lot. As a matter of fact, I just saw data the other day where we went back and did a comparison for a product that we released in 1999. That was lot 1. And they compared every lot from lot 1 to lot 29. And by Western blot and by IF, it's exactly the same. So if you're very careful about it, you can do it you can create that consistency by being very careful. The other way to solve that issue is by transitioning over to recombinant or monoclonal antibodies that are, by their very nature and production method, much more consistent from lot to lot. I hope that answers the question.
Thank you, Dr. Couvillon. Next question. Is the information on the antibody validation by DigiWest publicly available?
Okay, it's not publicly available at the moment, but with a paper to be published very, very soon, there will be a list attached in which we list approximately 300 of the antibodies that are working in DigiWest on the materials we were using there. These were several cell lines, tumor cell lines, and also primary human tissue. So this will be a starter. Then there will be a paper, and I was talking about the antibody validation characterization using the different species. while it has been published in a short way, there will be a proper paper on that, and that paper will include quite substantial information on the antibodies. And very obvious to say, if you would like to order kind of a DigiWest characterization from us, you do get each and every information on the antibodies and why they did get it into our list of antibodies. And this list of antibodies, of working antibodies, is available from our homepage as well. So there is not all information public at the moment. There's some public available, but there will be more very soon.
Next question. What would happen if the results of a DigiWest experiment called the specificity of a CST antibody into question?
I'll answer that one. So I think that some of the results that I've seen do that. And in those cases, what we've done is we've gone back and we've looked and we've reviewed to make sure that the antibody that we're selling is correct for the applications for which we've approved it. And in all the cases that I've seen so far, that's been the case. Not all antibodies are perfect, and I think that we do as much as we possibly can. And I think that the antibodies that we have on the market right now, I would stand behind every single one of them. But if we were to work with Markus and find one where the specificity was called into question, our typical procedure would be to do an internal review, notify the customers that there's maybe something wrong, and then work with those customers to make it right. Our worst fear is making a product that doesn't work the way that we say it does. So we take that kind of thing very, very seriously. And we appreciate when other platforms come in to cross-validate our antibodies, because it makes us more confident and it reveals any weaknesses that we have.
Thank you. Do you think more companies will start using platforms like DigiWest to routinely validate the specificity of antibodies?
Well, as a non-company person, I could try to answer that. It's not the only way where you can run kind of multiple Western blots. Other technologies have been developed over the years, and there are a lot of things happening. This really is for the good of the big antibody producing companies. So you do get much more information than you had over the recent years. And one of the reasons we really had to start that from scratch, that a lot of the antibodies which we bought initially were not good. We needed to have better information. So I think there will be similar technologies around, and there are around, and it will get better information on the antibodies you can buy.
Now, if you do run DigiWest assays, do you use pathway-specific panels of antibodies?
That's an interesting question. So yes, it is very interesting. Very often you've got an idea that a treatment or a tumor is of a certain type, and you might be interested in looking at 3 or 4 specific pathways which might be hit by a mutation or by a drug candidate you're interested in. And what we started, and we're real actively doing, is setting up panels of antibodies which we think make sense for analysis. Still, there is the big list, and everything which is on the big list can be chosen, but the panels are interesting to really have an idea how things might be changed. And we put quite a bit of work on setting these panels together.
Does this work for plasma serum?
Not really. So you've got the same problem with quite a few of the proteomic technologies. So the handful of proteins, or the 24 of proteins which make most of the plasma protein content, do interfere with analysis. So if you load plasma on the SDS-PAGE, you see, albumin mainly, some IgG bands, and a lot of the other things are not easily seen. If you deplete, things look totally different, and it can work like that. And we're actually thinking the other way around, so getting reactivity on serum plasma proteins and testing this reactivity, autoantibodies, however you might call that, on different tissues or on extracts from certain disease-causing agents. You can get information on that, which is a DigiWest turned the other way around, which is working and which is actually working very nicely.
Thank you. Next question. What is the cost of a study using DigiWest?
Okay, that's a tricky question. It really depends on the size of the study if you do that via our NMITT subsidiary. So running hundreds of antibodies on a dozens of samples gives you a price which is in the range of 6 to 10 euros. If the number of antibodies is getting lower, since it really is meant to test a lot of antibodies, prices can go up to, let's say, 12 or 14 euro per analysis. So that's a rough estimate. So if you're really thinking as a company and you're thinking and calculating the price of a Western blot, you will find out, okay, that is a bit cheaper than a classical Western blot. And if you go to a higher throughput, 200, 300 antibodies, it is much cheaper, and you really have to see, you don't need to care about antibodies. They're all here on our list.
We have some great questions coming in, but we have time for one more. And this is a two-part question. What type of mass spec are you using when you compare DigiWest and mass spec? I would think the advantage is to mass spec is you uncover unknowns, whereas DigiWest limits, although you can test many, to the antibodies you choose to test.
Okay, so the mass spec work was done in Bernhard Küster's lab in Munich. I think he was using an Orbitrap for the final analysis. So this really was high end. And you're absolutely right. So if you use mass spec, you have the possibility to look at everything, and you get an unbiased view, which never would be possible with the DigiWest. But if you get an idea where things are happening, the DigiWest might really be the right thing. And if you're looking at phosphorylation, it even might become more interesting. So not each of the peptides which does carry a phosphor group is easily detected using mass spec. Things are really getting much better in a much faster pace, but still having the DigiWest might give you complementary data. And mass spec and DigiWest I still see as something which is complementary. So another advantage of the DigiWest, but again you need to know what you're looking at, is that the amount of material is low, and it is still much lower what you would need for a mass spec analysis.
I would like to once again thank Dr. Markus Templin and Dr. Anthony Couvillon for their presentation. Gentlemen, do either of you have any final comments?
No, I hope that everybody found the webinar very useful today. And thank you to Markus for his excellent presentation.
Well, thank you, Anthony, as well.
Gentlemen, thank you so much. Now, if we weren't able to speak to you directly today, we'll be sure to follow you up via email. Thank you for taking part. I would also like to thank our sponsor, Cell Signaling Technology, for making today's educational webcast possible. Today's webcast will be available for on-demand viewing on our website through March 22nd, 2017. We will let you know when it's been posted, and we hope you pass that information on to any colleagues who couldn't take part in today's broadcast. Thanks for joining us. See you next time. Goodbye.